Hi, my name is Lara Darshane, and today I will be discussing everything that I have researched about the WI-38 cell line, from its origination back in the 1950s to how it's affecting everyday lives like mine today. So, let's get started. What is WI-38 cell line? The WI-38 cell line was the first normal human diploid cell line used to make vaccines. In the 1950s and 1960s, scientists relied on HeLa cells, which are derived from cancer cells, for virus research. While using HeLa cells were cancerous and unstable, often contaminating other cultures and raising safety concerns. In 1962, Dr. Leonard Hayflick and Dr. Paul Moorhead at the Wistar Institute developed WI-38 from the lung tissue of a 3-month-old female fetus that was electively aborted in Sweden. The sample came from a healthy donor, ensuring the cells were free of viruses and genetic defects. Hayflick discovered these cells were diploid, with 46 normal chromosomes, and could divide only about 50 times, a limit now known as the Hayflick limit. This finding revolutionized cell biology and aging research. Because WI-38 cells were non-cancerous, genetically stable, and pathogen-free, they became ideal for producing safe vaccines. During the late 1960s and 1970s, WI-38 cells were used to create life-saving vaccines for rubella, polio, rabies, chickenpox, mumps, and hepatitis A. The rubella vaccine, developed by Dr. Stanley Plotkin in 1965 using WI-38, remains the standard today. These vaccines have saved millions of lives and drastically reduced childhood diseases worldwide. While the use of fetal tissue sparked ethical debate, it's important to note that the abortion was not performed for research, and parental consent was obtained. Despite ongoing discussions, WI-38 is widely recognized as a pivotal breakthrough in science and medicine. It not only set new ethical standards for vaccine development, but also became a model for studying aging, genetics, and virology. Today, the legacy of WI-38 continues. Its vaccines are still protecting lives, and its discovery shaped the way we approach safe and ethical biomedical research. Now, let's move on and get into the biology involved. How do these cell lines replicate? Through a process of cell division. Cell division is the process through which a single parent cell splits to form 2 or more new genetic cells. It's essential for an organism's growth, repair of damaged tissues, and reproduction, especially in single-celled organisms. There are 2 main types of cell division: mitosis, which produces identical body cells used for growth and repair, and then there's meiosis, which produces sex cells, also known as gametes, with half the number of chromosomes, which is also used in sexual reproduction. For the actual reproduction of DNA before a cell divides, it copies all of its DNA during the S phase of interphase, ensuring each new cell receives a complete set of genetic information. The replication of DNA is controlled by the cell cycle and is divided into interphase, where the cell grows and prepares for division, and the mitotic phase, where actual division occurs. Interphase includes 3 stages: G1, where cell growth and normal functions; the S phase, which is where DNA replication occurs; and then G2 phase, where preparation for mitosis occurs and organelles and proteins needed for divisions are made. Along these stages of interphase are checkpoints which help prevent errors, ensuring that damaged or incomplete DNA isn't passed to new cells. Now, dividing further into cell division, we have mitosis. Mitosis ensures that when a cell divides, each daughter cell receives an exact copy of the parent cell's DNA, maintaining genetic consistency. Mitosis typically occurs in somatic cells, which are basically any cell in the body aside from sperm or egg cells. There are 5 main stages in mitosis. We have prophase, metaphase, anaphase, telophase, and cytokinesis. In prophase, the chromatin, which is the loose form of DNA, just going to set this aside, it condenses into a form the chromatin, it condenses into visible chromosomes. So each chromosome then has 2 sister chromatids, which are joined into a centromere. The nuclear envelope breaks down and spindle fibers start forming to help move the chromosomes. During metaphase, which you can see in the second image here, the chromosomes line up in the center of the cell along the metaphase plate. So we have all the chromosomes, they're lined up here now. This isn't a physical liner plate, but a visual one where you can see the chromosomes line up. The alignment makes sure that each new cell will get an equal and identical set of chromosomes. So next, in anaphase, the microtubules, they shorten and they start pulling the sister chromatids apart and then moving them towards the opposite poles of the cell. This separation is what guarantees that each new cell will have a complete set of chromosomes. And then during telophase, the microtubules break down since their job is done, and two new nuclear envelopes form around the separated set of chromosomes. So here in this last image, you can see the new nuclear envelope form, and the cell is starting to complete itself. The chromosomes start to uncoil back into chromatin, and the cell begins to look like it's splitting into two. And then finally, in cytokinesis, the cytoplasm divides to create two separate cells. When mitosis is complete, we end up with two identical daughter cells, each with the same number of chromosomes as the original. For humans, that's 46. Now that we know how these cells divide, we need to know how to sustain them. Now, to keep these cells healthy, researchers have to grow them under very specific conditions. They're usually grown in nutrient-rich liquid medium, like Eagle's Minium Essential Medium, which provides all the vitamins, salts, and amino acids the cells need. This medium is also mixed with about 10% fetal bovine serum, which adds extra nutrients for growth. The cells are kept at 37 degrees Celsius with about 5% carbon dioxide, which mimics the conditions inside the human body. Because the cell grows and fills up the container, they need to be subcultured, or split, before they become overcrowded. This helps keep them healthy and dividing normally. All of this has to be done using aseptic technique, meaning everything must be sterile, so no bacteria or fungi contaminate the cells. For long-term storage, WI-38 cells are frozen in liquid nitrogen at around minus 196 degrees Celsius, using a protective chemical called DMSO. This freezing process lets scientists store the cells for years and then thaw and regrow them when needed. Finally, researchers always keep track of the passage number, which tells them how many times the cells have been grown and split. Since WI-38 has a finite lifespan, this helps ensure they're using healthy, stable cells for research or vaccine production. Let's now move on to some of the considerations and controversies that have been created from these cells. The WI-38 cell line was created in 1962 from the lung tissue of a 3-month-old female fetus that was electively aborted in Sweden. It's important to note that the abortion was not performed for research purposes. It was done with personal reasons, and afterward the tissue was donated for scientific use. Back then, there wasn't as many ethical guidelines about using human tissue in research as there are today. Because of that, when WI-38 started being used to make vaccines, some people began asking serious ethical and moral questions, like whether it's right to use medical products that come from aborted fetal tissue, even if they're used to save lives. This sparked religious and social debates, especially among some religious communities, such as within the Catholic Church. Some people felt that using vaccines made from fetal cells was morally wrong because of their origin. However, over time, many religious leaders and ethicists have agreed that using these vaccines is acceptable, since the abortion was not performed for research and the benefits saving millions of lives are enormous. Even the Vatican had stated that when no alternative vaccines were available, it's morally permissible to use them to protect individual and public health from a scientific standpoint. The original fetal cells from 1962 are still used today. They are replicated in laboratories, so no new fetal tissue is collected or needed. That means the same cell line has continued to help produce safe vaccines for decades without repeating the original procedure. Despite the ethical debates, WI-38 has an incredible impact on global health. It has been used in vaccines that prevent diseases like rubella, rabies, chickenpox, mumps, and hepatitis A, and those vaccines have saved millions of lives worldwide. Because of how much the cell line has changed the lives of millions, let's take a deeper dive into what the public health policy has been like since. The WI-38 cell line completely transformed the landscape of modern medicine, not just scientifically, but politically and ethically. From a public health standpoint, it was revolutionary. Using WI-38, scientists developed safe and effective vaccines for rubella, measles, mumps, rabies, chickenpox, and hepatitis A, diseases that once caused millions of infections and deaths every year. In fact, the rubella vaccine alone, which was made possibly by WI-38, prevented an estimated 11 million miscarriages and congenital birth defects worldwide. That's an incredible public health success, and it helped prove that human cell lines could produce safer vaccines than older animal-based methods. Because WI-38 cells were free of animal viruses and genetically stable, they raised new safety standards for vaccine production. This led governments and health agencies, including the World Health Organization and the U.S. Public Health Service, to invest more heavily in vaccine research and establish strict manufacturing guidelines to ensure vaccines were clean, safe, and effective. But beyond the science, WI-38 also pushed the world to rethink the ethics of biomedical research. Since the cell came from the human fetal tissue, they prompted new questions about consent, privacy, and the moral use of human biological materials. In response, the U.S. and many other countries began developing ethical systems for research involving tissues. This ultimately led to the creation of the Institutional Review Boards, or IRBs, which oversee research to make sure it's done ethically. It's also influenced the National Research Act of 1974 and the Belmont Report in 1979, documents that shape modern bioethics. They introduced three main principles: respect for persons, meaning people must give informed consent; beneficence, meaning researchers must minimize harm; and justice, meaning the benefits and risks of research must be shared fairly. So in many ways, WI-38 didn't just save lives through vaccines, it also helped establish the ethical and legal foundation for how we conduct biomedical research today. It set the tone for how scientists collect, store, and use human biological materials, ensuring that research is not only innovative, but also ethical and transparent. Let's now talk about the medical impact it's created. WI-38 was crucial towards vaccine developments as it allowed scientists to grow viruses in a safe, human-based environment. Because these cells are normal and non-cancerous, vaccines produced using WI-38 are safer and more reliable than those grown in animal cells, which can carry additional risks. Some of the most important vaccines developed using WI-38 include rubella, measles, mumps, varicella, rabies, and hepatitis A. For example, the rubella vaccine, specifically the RA27/3 strain, was made possible using the WI-38 cells. These vaccines were not only effective but also free of contaminating viruses and more reproducible than older methods. The WI-38 cell line has had a massive impact on global health. The rubella vaccine alone prevented millions of cases of congenital rubella syndrome, a disease that can cause serious birth defects. By supporting massive immunization programs worldwide, WI-38-derived vaccines have reduced disease incidence and mortality, saving millions of lives and preventing severe illness. Because vaccines grown in WI-38 are safe, stable, and consistent, public health authorities could trust immunization programs, which was key to their success. Now, I want to transition into something a little bit more personal. When I first learned about the WI-38 cell line, it seemed like just another scientific breakthrough, something that existed far away in a lab decades ago. But the more I understood its impact, the more I realized how directly it had shaped my own life and the lives of the people I love. For instance, I never had the chickenpox, something that used to be almost unavoidable for kids. The reason is simple. I received the chickenpox vaccine, which was developed using a WI-38 cell line. It's easy to take for granted, but when I think about it, how many children used to get seriously sick or miss weeks of school because of chickenpox? It made me appreciate just how powerful this discovery really is. But WI-38 impact goes beyond me. It also touched my family in a much deeper way. My dad had 2 strokes in 2021, and ever since then, his health has required constant care and protection. For someone like him, even a simple viral infection could cause serious complications. Knowing that vaccines developed from WI-38, like rubella, measles, mumps, and hepatitis A, help protect him from preventable diseases brings me comfort. It means he can focus on recovery without facing additional health risks that could have been devastating. WI-38 isn't just a specific milestone. It's a reminder that medical discoveries have real lasting effects on everyday lives. It represents how decades of research and innovation continue to protect us, even when we don't realize it. For me, it's proof that science isn't just about experiments and data, it's about people. It's about giving families like mine more time, more safety, and more hope. Now, my stance, when it comes to the use of human-derived cell lines in research and medicine, I believe it's one of those areas where science and ethics have to work hand in hand. The WI-38 cell line, for example, was developed from fetal tissue in the 1960s, a time when ethical standards and consent procedures weren't as strict or as transparent as they are today. Knowing that makes it complicated. On one hand, this discovery has saved millions of lives through vaccines and medical advancements. On the other hand, It raised important questions about how we obtain and use human biological materials. My ethical stance is that using human-derived cell lines can be acceptable and even necessary, as long as it's done with complete transparency, informed consent, and respect for the people whose cells are being used. The benefits to humanity are enormous, preventing disease, advancing treatments, and improving quality of life. But those benefits should never come to the expense of human dignity or autonomy. I think the WI-38 storyline I think the WI-38 story reminds us that ethical reflection should grow alongside scientific progression. It's not enough to focus on what science can do. We also have to ask what it should do and how it can be done responsibly. For me, this balance between innovations and ethics is what makes medical research truly human. It's about saving lives while honoring the life that made the research possible in the first place. And that's all this. And these are all my sites that I use in all my research for today. And I hope you enjoyed this, my research topic. Thank you.